So hi, hello again, and today's topic, prokaryotes. These are bacteria. So I'll make a drawing of a bacterial cell today. Of course, we're going to label it again. You already know that. And of course, we're going to also have a look at bacteria under my microscope. Yeah, and so far you have already watched a few videos on cells. I hope that you did that. And plant and animal cells, those were so-called eukaryotes. Yeah, so these are cells that have all of those cell organelles. But prokaryotes, the topic that I'd like to talk about today, well, those cells are quite a bit different. Well, not quite a bit. I mean, there are always some similarities, but they are a little bit different. And that's why I'd like to talk about those cells today. And as you can see, yeah, I have again prepared a whole bunch of labels here and descriptions, and we're going to add them to my drawing later on. So I carefully have to push everything to the side again. And let's start off by making a drawing of a prokaryote, of a bacterium. And there are certain things that I recommend, like for example, you always start drawing the cells, or pretty much anything that you draw, the same way, because this way you do not forget anything. So I'm going to start drawing a prokaryotic cell, and I always start off by drawing the so-called the cell membrane. In this case, I'm going to make the cell a little bit longer in shape. So this here is the cell membrane. I will be adding again all of those labels later on. Just going to add an arrow. Yeah, and then later on I'm going to add the label. Well, inside of the prokaryotic cell of a bacterium, you have the cytoplasm. Yeah, so I think that's just the liquid in here. I can just make an arrow over here. This here is the cytoplasm. Also, eukaryotes have this, of course. And then very important now is this prokaryotes, they do not have a nucleus. They still have DNA. But this DNA is present in the shape of a ring, a circular ring, floating inside the cell without a nucleus. So I'm going to draw the DNA now, and because it's ring shaped, look, it is a large loop of DNA. Okay, and then you look over here, it closes again. Yeah, so that's basically simply the DNA floating around inside the cytoplasm. There is no membrane around it. Yeah, so there is no nucleus, no cell organelle around it. So I have to make a label here as well. And there are also ribosomes present. Those ribosomes are also floating around inside the cytoplasm. So I also have to label those, the ribosomes. Yeah, and in many cases, bacterial cells also have a cell wall. Now be careful. You do remember that plant cells also have a cell wall, and that is an important distinguishing criterion between plant and animal cells. Plants have a cell wall, animal cells do not. Yeah. Bacteria also have a cell wall, but they're not plants, of course. I mean, the cell wall is made of a completely different substance. Well, the only thing that it has in common is that the function is similar. It also prevents the cell from bursting. But what I'm going to do now is I'm going to draw a cell wall now on the outside here. That's a little bit of a problem because I'm going to go over now the labeling lines here. That's not so good. Okay. Yeah, so you see that this line over here does not even touch the cell membrane anymore properly. Yeah, so I'm not going to continue drawing it all the way. But you get the idea that this here, different color. This is the cell wall. Okay, so actually, strictly speaking, it's the whole thick part here. But if I color it in, then we're not able to see the cell membrane again. Yeah, it's a little bit difficult to draw using those felt tip pens, and for this reason I always recommend that you use a pencil. Yeah, and that is almost it, because in the cytoplasm what we have is we also have very small ring-shaped DNA molecules. Now how am I gonna draw those? I'm gonna draw them as a little circle here. It looks like a vesicle. It's not a vesicle. Okay, these are not cell organelles. Yeah, but this is referred as a plasmid. A plasmid is a small ring-shaped DNA molecule. Okay, and we're actually pretty much finished for right now. So what I'm going to do now is I'm going to simply add the labels. Maybe if I forgot something, then I have to add it later. So circular DNA, that is this one over here. Okay, that is fine. The cell wall, this is this thick black structure over here. It's the cell wall, which prevents the cell from bursting. After all, bacterial cells are also under pressure Ribosomes, we got those. They make proteins. Okay, eukaryote. Wow, no, that is a different label. It's for a comparison aspect. Look at this. This is also not, doesn't belong here. And prokaryote. Of course, the whole thing here is a prokaryote. We already got the label here. Plasmids. Okay, here we go. That is a plasmid. The cell membrane, of course, is here, and the cytoplasm. See, I got everything here that I needed. Okay, yeah. So this is essentially a prokaryotic cell. There are many different shapes that prokaryotic cells can have. Some are a little bit longer, like the one that I made over here. Others are round and circular. Still others are spiral shaped. Some are even able to move. Yeah. So the diversity is also quite high. But there are certain basic structures that all bacterial cells have the same. Okay, and then just for the fun of it, I'd like to show you now a short video. You probably have already seen it in one of my previous videos here, but these are spiral-shaped bacteria and they're moving. Yeah, so some of them are even moving around and whizzing around. Yeah, and you can see that they're pretty small actually. Yeah, they just look very dark in this case. And they're also difficult to see because they're transparent. But my microscope is able to make them darker on a bright background, so I'm using special optics. But I have to tell you, bacteria, because of their relatively small size and because inside the cell you do not see any cell organelles, for this reason they're kind of difficult to observe as well under the microscope. But I just wanted to show them to you that yeah, they look quite different to plant and animal cells, of course. But they do all of the functions of life nevertheless. So let's move back here. Here we go again. Okay. So what I'm going to do now is I'm going to now, yeah, add, yeah, all of those descriptions. I already talked about those shape of DNA. That's also for the comparison table. Yeah, additional genetic material. These are the plasmids. I'll add them down here. Okay. Then they make proteins. I should have maybe made the label a little bit smaller here, but the ribosomes are responsible for making proteins. I think, you know, what I would suggest is that you don't add the text now to the drawing, but that you make a separate table. You write down ribosomes and next to it you write down they make proteins. Okay, this is the genetic material. That is the DNA. Okay, look, that's a little bit chaos. Nucleus present. No, that's again for the comparison. Okay. Contains enzymes. That is the cytoplasm. The cytoplasm contains enzymes. Yeah, here we go. Yeah. Later on at the end of the video, I'm going to arrange this much more nicely. You see, you cannot even see it properly because I'm in the way. This one also doesn't belong here. It prevents the cell from bursting. That is, of course, the cell wall. I'm just going to put it up here. And it controls what goes in and what goes out of the cell. This is the cell membrane. Okay, so we not only labeled everything, but we also added now the function, yeah, of the different parts of a prokaryote, of a bacterium. Okay, so yeah, I'm going to move on now, and I'm going to now do a comparison table here, and for this reason, I'm going to clear everything away again, and I'm going to flip this piece of paper around, and we're going to make a comparison table now. Maybe not so good because yeah, there is a little bit of you can see a little bit here. Doesn't matter. You always have to add a title. The title is Prokaryotes versus Eukaryotes, and yeah, I'm going to make a table over here. Always use a ruler when you make a table, okay? But I'm allowed to do it like this, okay? Because I have to quickly make a video here for you. Yeah, and I'm going to label, of course, the headings here. Aspect. The aspect is the thing that you want to compare. Over here you have a eukaryote, and over here you have a prokaryote. Remember, the eukaryotes are plant and animal cells. And the most important difference is always the question of whether a nucleus is present or not. Okay, so this is the most important. You know what? I'm going to use scissors now. Look, because I need to make sure that you're able to read this properly. Okay, here. So is a nucleus present or not? That's the most important one. Why is it the most important one? Because the word prokaryote means before nucleus, and eukaryote means a true nucleus. Okay, so eukaryote means those cells have a true nucleus. So is a nucleus present in a eukaryote? Yes. Okay, and a prokaryote, no, because you remember that the DNA is floating around freely inside the cytoplasm and there is no nucleus around it. Yeah. The second thing is that the shape of the DNA, that's another important difference. Okay, let's put it in here. Okay, how does the shape of the DNA in eukaryotes look like? It is linear, okay? Linear. So basically you have several linear pieces, several actually, okay? Like for example, humans have 46, they're like the chromosomes, they're open-ended. And in a prokaryote we have a round circular. As I mentioned, look, and it closes back on itself again. So the DNA, the shape of the DNA is different. And the last important difference is the size. And the size of a eukaryote, it's larger. Larger and smaller. So I'm going to also tell you the size. It's approximately 10 micrometers long across. I'll be talking about the size and size calculation in another video, but that's this U with a long line over here. That's the sign of micros. It's 10 millionths of a meter, and smaller is 1 micrometer. So it's basically a millionth of a meter. So if you take a millimeter, which is very small, a millimeter, and if you divide it into 1,000 times, then you have the size of a prokaryote. If you divide it into 100 parts, then you've got a millimeter, then you've got the size of a eukaryotic cell. So both are small, but prokaryotes are significantly smaller. Okay, so this is an important comparison table between prokaryotes and eukaryotes. Yeah, and something that I did not include over here, but you should know that contains mitochondria. Eukaryotes, yes. Prokaryotes, no. Because all of the other structures that we've seen in a eukaryote, like mitochondria, chloroplast, rough endoplasmic reticulum, Golgi body, all these things are not part of a prokaryote. Yeah? So what am I going to do now? I think I'm going to, you won't believe it. Look what I brought here. It's my breakfast. Why am I eating breakfast right now? I love yogurt and cereals. Yeah. Really good, because of course in yogurt you have billions of bacteria. Yes, you add bacteria to milk, and then the bacteria, so-called lactic acid bacteria, what they do is they start to divide and reproduce, and they convert the milk to yogurt. And I think it's a very nice thing to put under the microscope. So let's see if I'm able to find some bacteria in this yogurt. And yes, sure I am. Well, and in order to look at some yogurt bacteria, we need to clean the table again a little bit, get everything out of the way, what I don't need. And let's move this up a little bit. And here is my microscope slide and a cover glass. And big bacteria are difficult to see under the microscope. Okay, so you have to take a very small amount. So otherwise the bacteria are going to move all over the slide and they're difficult to focus. So what I'm going to do now is I'm just going to take here the back of my spoon here. I'm going to dip it carefully into the yogurt. You see? The small amount here should be enough. Okay, I'm going to carefully put the spoon again over here on the top. Let's move my yogurt away. Cover glass goes on top. And look how it spreads. And you know what I'm going to do is I'm going to press even further against it to really make it flat and thin. I'm also going to refocus it a little bit so that you see it better. Yeah. It's difficult to see, so there's almost nothing on here. Very thin film of yogurt, and I'm going to put it under the microscope. So let's switch over to the microscope view, and let's turn on the lamp of the microscope. Yeah, so here we go. Let's open it a little bit further, and let's hope for the best. Let's see. Let's move this also a little bit out of the way here, and here we go. And okay, there's a fiber here right in the middle. Yeah, that's the magnification is not large enough. And what I'm going to do now is I'm simply going to go up with the magnification a little bit further, and then I'm going to switch over to phase contrast. So maybe I should do this right away. Let's switch over to phase contrast. I have to add the correct filter. So, and let's open this here and turn up the light. So, and yeah, we have to now try to focus everything correctly. And then if we're lucky, we're going to find some bacteria. And all of those dots that you see, yeah, maybe there are bacteria in there as well. Look at this blue gigantic fiber. Look at this blue gigantic fiber. It's just some dust that was on the microscope slide. Yeah. There are a lot of milk proteins in here. Yeah. But between here, the fiber, this blue fiber is huge. Yeah. But look here carefully when you see this here. Do you see those dots right now in the middle? I'm going to point towards it. Those dots that you see over here. Yeah. Yeah, all of those tiny, these seem to be bacteria. Okay, and you see that they have a different shape than the ones that I just showed you in the video. And here, all of this other white stuff that you see, these are this coagulated milk protein probably. But here, everywhere, nice tiny bacteria. Yeah. Here, if you look carefully, you actually see that I'm floating away in the liquid. Yeah, but there are many of these yogurt bacteria in there, and they produce lactic acid, and this is responsible for converting the milk to yogurt. In my other, you probably already know that I have also another microscopy YouTube channel, and this is I also made videos there where I put yogurt under the microscope. Yeah, here there are many bacteria. That's nice. I include a link, and then you can look at those videos as well. Look, many yogurt bacteria. Here, the round things. These are air bubbles over here. So in other words, when I eat my cereal, when I eat yogurt, then of course I eat a lot of bacteria, and those bacteria, they most of them are digested and are killed by the acid of my stomach, and some of them actually are able to survive. They end up then in my intestine, and they continue to live there because that's a normal part of our digestive system. So so you've now seen also yogurt bacteria, also known as lactic acid bacteria, and there are several different types that you have. That they put into yogurt. You can also buy them in a powdered form if you want to make your own yogurt, but you don't have to do that. All you do is you add a little bit of fresh yogurt to some milk, and then you have to wait for a day. You keep it stored at room temperature, and then the bacteria starts to grow and reproduce, and then you have the next day you have yogurt. You look very carefully, you're able to see them float a little bit. So this is basically something I wanted to show you. Okay. Today's topic were the prokaryotes. Here it is again. Those prokaryotes have a much simpler cell structure than the eukaryotes, are much smaller, and bacteria. And now you also know that not all bacteria are bad, but as a matter of fact, some of them are very important for breakfast. So we eat them every day. I like to eat yogurt every day. So, but no eating in the lab, no eating in front of the camera. I'm going to leave it at that now. Hope that you were able to follow well what I was talking about today. I wish you all the best, and see you around in the next video. Bye-bye.